The CUROP program is invaluable for what we're trying to do here. So it's a project that we're sponsoring for, it's like a 10-week project, but it's much more than just a 10-week project. 10 weeks initially, but then the students are involved with developing a website and presenting their work, both a poster and also this conference in Boston. So it gives an opportunity for a kind of more holistic idea of a project. So a lot of CUROP students might come in and just work on an individual project. But what we try to do here in the School of Biosciences, working on this iGEM project, is to have the students, and we have 7 students in total, 4 of which are CUROP funded. We have to have them working together to get a lot more independence within their work. I think a lot of students, when they finish their degree, within a degree program, there is a lot of babying, let's say, if you will. So students are handed things to do and they do those things. But within the CUROP program and within the project the students are working here, they're a lot more independent. So their learning skills enable them to be independent in their work, which will hopefully benefit them when they leave this lab environment and go on and do whatever they do. Really, this project started in March this year, and we have a discrete 10-week period now in the summer, but it will go on until October. So it's really 6 months that I'm working with the students, and I can see a definite change in their personalities and how they interact with each other, certainly. So a lot of the students came in to this project, which is based on plant science, without any knowledge of plant science whatsoever. And now they are developing their skills and interests and expertise in that area. And by the end of the project, as I say, they'll be presenting it in front of up to 1,000 people in this conference in Boston, so they'll have to have a good level of expertise in order to do that efficiently. One of the strengths of the CUROP is that it's relatively straightforward to apply for the funding. So you come up with an idea, you put together a relatively straightforward application, and then you hear a few weeks later whether it's successful or not. So it really takes a lot of burden off significant grant writing and things like that. It makes it relatively straightforward, but having the interaction with the students before you apply as well can be beneficial, because you know that what you're going to get, and when you get the money, it can be really beneficial for both myself, for my research, and also the student development, I think.
So what we're doing with the project is we are trying to create an effective pesticide against aphids. Currently, the pesticides that are used are chemical pesticides, so they're quite unfavourable because they can be non-specific and aphids can develop resistance to them. Aphids, they're vectors of viruses, which means they can kill many plants, and therefore, especially in countries like China, they're massive crop pests. So what we're trying to do is we're trying to make a GM approach to kill them. I have multiple sort of roles. I've got a lab sort of based side because I'm a geneticist, where we are actually doing experiments to try and quantify how much RNA we can produce and sort of measure the toxicity of it, so the effectiveness of it. But also I have to design a website where we can document all of our sort of research and all of our findings, and that will be presented in a big conference in Boston, so an international conference with loads of different teams. And then another part of it is that I'm mathematically modelling what we might expect to happen both in the lab and experiments, which you have to sort of estimate the sort of toxicity of each of our RNAi constructs, and then also what would happen in the field where you've got multiple other factors like predation and migration, which isn't obviously present in the lab.
So at the moment we're trying to get bacteria to produce the parts that will hopefully kill the aphid later on. So they come in little parts and we have to kind of put them all together. So at the moment we're trying to get them all into one thing, but it's proving a little bit tricky. And then once they're all in the same plasmid, we can then inject the bacteria into the plants and hopefully get the plants to express the DNA. I've never done this before. Working with the plants is really fun. I've never done a lot of work with plant biology before. The infiltrations are really fun, where we get the bacteria and we kind of use a syringe to get the bacteria inside the plant, and you can see the bacteria spreading across the leaf, and that looks really cool. Everyone, well, a lot of people don't like GM. They think that it's dangerous and that eating GM crops is bad for them, bad for the environment. And a lot of people are misinformed about it, so it was really cool to see people learn a bit more about what GM actually is and how it could really benefit us, because pesticides aren't good either, but I think people are quite set in their ways. So it would be really good to see more people be open to genetic modification in crops and in other areas because I think it could be really beneficial. So I mean, ideally it would be great to see this idea and more GM ideas implemented in reality, although I think it's going to be a really long time before that happens.
Yeah, so I've got a bit of a mixed role. I'm doing human outreach stuff, but I'm also working in the lab as well. So day to day I do get involved in all the lab work, but also I help getting involved in organising outreach events and running those events and things like that. So we just did an event where we went to TechniQuest and we took demonstrations with us. So we did strawberry DNA extractions and we did plant infiltrations as well to try and teach younger children about what DNA is and what you can kind of do with genetic modification and introduce these ideas to them.
Doing a CUROP project has been completely invaluable. It's been an incredible experience because you don't really get the opportunity to do it otherwise. So being able to do a funded project, which is, at least in part self-managed, especially with the way we're doing it as a team. It's developed my scientific ability, and obviously I would never have done mathematical modelling or website design as a geneticist, so it's given me the opportunity to learn many transferable skills.
I'd heard of CUROP before last year, and it seems like a really good opportunity to experience research over the summer and give you kind of a chance to experience what it's like for our project's 10 weeks. So it's a long time, so you get used to kind of what it's like in the real world to do that kind of job.
We have got some results scientifically in terms of the expression. So basically we have to measure protein output, and we've got some images of that we can show. In this case, we've got a green fluorescent protein, so it makes the leaf veins where the aphids drink glow green. We're also testing stuff that'll make it blue or red. We've got some data on that already. We've mathematically modelled it to show what would happen in the lab. And what we hope is that if our RNAi constructs are toxic, it will sequentially deplete the aphid populations, or at least decrease the growth, depending on the parameters that we use. So we have 2 lab models, one of which suggests that it will effectively control aphid populations so that they'll eventually die out, and another in which that it will reduce the aphids' birth rate so they're not as much of a problem, but the populations still boom after a while, which is obviously unfavourable but potentially realistic.